your exit ticket question from Friday. And then we will finish looking at types of passive transport and do that with respect to the lab that you're going to do. Today, you're going to get some information for writing your introduction. And you should be able to write the first bullet points on your introduction after this. Tomorrow, I will show you the process that you're going to use, and you'll be able to, you're going to write your procedure because you are going to make some decisions about what you're going to be testing. I will not be saying you're going to see if this happens. You'll be looking at what you know and making that decision on your own. Wednesday, you will set that up in lab. And Thursday, you will collect your data to see if the things went through the membrane that you thought would or that you didn't think would, and get your evidence for that. And then Friday, your job will be writing that claim, evidence, reason, conclusion. So we'll spend some time talking about that as well during the week at some point. But first, your exit ticket. Your exit ticket asked why endosymbiosis was a theory and not a law or hypothesis. And I didn't grade the whole exit ticket because after reading most of them, I'm like, clearly we don't understand this. And it's not that you didn't understand endosymbiosis, it's that you didn't understand law, theory, hypothesis, and proof, which if you say that in the lab report, I will send it back to you. You will not discuss proof in science. It doesn't happen. Proof is what happens in a court of law when they say, well, we found your fingerprints at this place, so you did it. It's a one-time thing. It doesn't happen in science. So what we do know in science, here's the evidence that we have. And if you need to go back and change this view, I will grade it later tonight. If you don't change it, you're not going to get credit for it because you can't say, well, we don't have any proof, or we didn't, it was crazy, answers like that. So what we saw is in different cells, we saw that there were 3 different organelles that had a double membrane. What were they? What were they? Who were the 3 that had double membranes? We saw them in plants and animals and some prokaryotes. We did, or some protists. We did not see them in prokaryotes. What are those 3 organelles that have double membranes? Go. Yes. And what? Yeah. So there was a wondering about how that possibly happened. And when you look at the timeline of when we see fossilized cells of different types, we saw that there were protists before multicellular plants, and we saw that there were prokaryotes before that. Some of the prokaryotes were able to produce energy from chemicals that they took in, or and oxygen. Some didn't even need the oxygen. They got the oxygen from other chemicals, from carbonates and such. We also knew that there were cyanobacteria, blue-green algae before those things. And so an explanation that works really well is that the nucleus formed when a cell began to have some, what are these called? We have these little pouches that form. We have enfolding that happens. And as this bit of membrane folded in, sometimes it went around the membrane and then broke off. Okay? And so that's kind of how the nucleus formed, because it would encase some of the DNA. At the same time, we learned that the membrane went around the nucleus and formed from enfolding, then aerobic bacteria here might have been engulfed. And again, because it already had a membrane, and then the other cell membrane formed around this, we had a double membrane. If that happened to be a mitochondria, then that cell now had its own way to produce energy. The cell provided protection for the new mitochondria. The mitochondria provided energy for the cell. If that's all that cell engulfed, it would go on to replicate over and over and over again and be what we think of as an animal cell, which is heterotrophic, cannot produce its own food. If that cell, maybe later, engulfed cyanobacteria, blue-green algae that had chlorophyll in them, then that cyanobacteria became a chloroplast, and we had a plant cell with both the mitochondria and the chloroplast. So this is an explanation of what happens. We have plenty of evidence for this. We've never seen a eukaryotic cell that this wasn't the case, that they're chloroplast if they have one, and their mitochondria and their nucleus don't have a double membrane. So I'd say that's plenty of evidence. So you don't need to say, well, there's enough evidence, so it's only a theory. And only a theory is not something you say in science. We have a lot of evidence, a lot of information, facts, if you will, and the endosymbiotic theory is an explanation of how that happened. Do you have questions about that? Anything going on there? So if you need to go in and clean up that statement on your exit ticket, please do, because they weren't so correct on the turn-in. Also, please remember on Friday to turn in whatever it is you need to turn in. A few of you in this class and others did not, so not a good thing. So with that, let's look again at passive transport that we started looking at. We're going to finish up how things get in and out of cells without the cell expending any energy today. And then tomorrow, we will look at how you will test for that in this lab. You're going to be making a little replica of a cell out of some dialysis tubing. We talked about that, which is what kidney patients have when they go to have kidney dialysis because their kidneys aren't working. That membrane filters out the bad stuff and puts in the good stuff. And you're going to use that to test what might move in and out of a cell membrane by a concentration gradient. And you do remember concentration gradient, right? Happy birthday, by the way. You remember concentration gradient, yes? Here's that again that you're looking at. And glasses of iced tea. You don't have a concentration gradient in the first two. You just have one that's got more concentration than the other. But this guy in the end, you have a high concentration at the bottom where this 12 tablespoons of sugar that you've dumped into your iced tea builds up. And then it becomes less concentrated as it moves throughout. We did a demo where I took some dye in a beaker, and this one was from your class. At first, how did this look last week when I first put this in? How did it look last week at first? How did it look then? Yeah. But I didn't stir it, I promise. But over time, because the particles all move by diffusion, the dye spread out, and now we have a very uniform looking solution. Same thing happened here. This one was from 6th period. Actually, by 6th period, yours was pretty well diffused through and looked pretty uniform. And so this is more what your lab is going to look like. You're going to take a little piece of tubing, and you're going to take a beaker. The tubing you're going to fill with something. The beaker you're going to fill with something. You're going to have distilled water. You're going to have pretty concentrated solutions of glucose, starch, protein, and a new one, salt. And you're going to decide, you want to see whether things move in or out of the cells. And you're going to decide that on your own. You're going to need to do all 4 of those. We also have food coloring, so you can put that in one place or another as well and see what that does. It's not a biomolecule, but sometimes fun to look at. So you're going to put some combination of those solutions inside or outside of your dialysis tubing. Okay, so if you put water in the dialysis tube, you would not put water outside, you put them in a solution. Usually suggest you put the other solutions inside. You're going to let them sit overnight, and you're going to test them on Thursday to see what's going through the membrane. You're going to use the same test you did for the biomolecules. So if you want to test, if you put sugar inside and you want to see if it moved out, you're going to use a Benedict's test. You're going to take some of your outside solution, put some Benedict's solution with it, and figure it out. If you put a lot of salt solution inside and you want to see if the sodium chloride moved out, you're going to use a new test. It's silver nitrate. And if it turns cloudy, then silver nitrate moved out. And then salt moved out. That's kind of what we're looking at. We need to look at how some of those things happen a little bit more. So here's a little bit more on passive transport. This is new stuff for you. Passive transport, you can date this November 2nd, so you can find it again. As you will remember, passive transport is when there is no energy needed from the cell for this to happen. It's driven completely by concentration. There's a lot of salt in the solution inside the cell. It will move out just because of the concentration that's higher there. The cell wants to go to equilibrium with its surroundings. Substances will cross a cell membrane by moving down a concentration gradient. So you go from high concentration to low concentration, just like a truck at the top of a hill. If you take the emergency brake off, it moves from the high spot to the low spot. No energy is needed from the cell. This is passive transport. This is happening to all your cells as you are hanging out right now. You had anything for breakfast, you're digesting it, and it's moving into the cells to give you the things that you need. Providing you some energy mostly. Protein that you ate is broken down into amino acids. The amino acids are transported around and go into the cells that need them. Water moves in and out of your cells. Oxygen will move into your cells. Carbon dioxide will move out as your mitochondria break apart sugar to give you energy and produce carbon dioxide. Anything they're not making sense to you. A whole lot of something on one side of the membrane, less than the other, it's going to go from a lot to less. Tries to even out and reach equilibrium. Okay. And this concentration gradient works through air, water, solution, or the cell membrane. So just like in the dye that we put in our beakers of water, there's a high concentration where the dye first fell into the water. And then it distributed itself. It wanted to equilibrate throughout the water. Now remember your structure of the cell membrane that you labeled the other day. Look at that and look at it. That might be helpful. That nice color picture you had of the cell membrane. And you'll remember that there are different parts in the cell membrane. Some of it was just lipid, the phospholipid part. And things can move through that if they happen to be soluble in fat or very small. Then you have the other proteins that help with things. We're going to talk about some of both of those. And it's all of those things that make the cell membrane what we say is selectively permeable. Selective means choosy, so it doesn't let just anything in and out. And permeable means that the substances can move in and out. It will let things in and out, but it's choosy about the substances that can penetrate the membrane. So the cell membrane is selectively permeable because if something is not the right size, shape, chemical makeup, it will keep it out. There are a couple of types of passive transport. We're going to look at three. The first is simple diffusion. Simple diffusion, the particles of a substance can move through air or water or a solution or in and out of the cell by the concentration gradient. That's what would happen here in your beaker of water and dye. So your first three bullet points in your introduction of your lab are about types of transport. I'm giving you everything you need right here to write them. You should not be having to do a lot of random Googling and writing crap that you don't understand. You should be able to just process what you have right here to do those things. Simple diffusion. Particles of a substance move through air, water, or a membrane. They're driven only by that concentration gradient. You've heard concentration gradient enough times by now that you should be able to see what it is. This does not use the membrane proteins at all. Membrane proteins are not, other than being maybe an open channel, they don't, nothing happens here. If the particles are soluble in lipid or made of lipid, they can move easily through that membrane. And examples of those would be steroids, which you do produce on your own. Not all steroids are bad. They're only bad when you take high concentrations of those that you don't have or think you need. But your body does produce some, and they are very, very important. Hormones also. big one that we all use every day is insulin. And that one you should take if you need it. And then tiny molecules like O2 and CO2 can pass through as well. So again, this information should be all that you need to write the first 3 points in your introduction. And I would suggest you do that tonight. The document is there in classroom. Now I'm going to turn it in today. And that you'll come back and turn this in when you're done writing your conclusion sometime after Friday. But you will turn this in classroom. So this is the first type of transport that will be happening. In your lab. If a substance that's trying to get into a cell is not soluble in the lipid or native in the lipid, they need some help to get through the membrane. And we do need some things that aren't soluble in lipid or native lipid. We need proteins, we need glucose. And so the help or facilitation that they get is provided in another way. Simple diffusion type 1, stuff goes right through the lipid, and that would be a steroid, a hormone, O2 or CO2. This will bring us to our second type of passive transport. Which is facilitated diffusion. And a number of things can get in this way. This is where the actual proteins in the membrane. So here you see the proteins in the membrane. These are the membrane that you built. And they have a channel through the middle of them, or a passageway, where things can go through. So the proteins in the membrane help or facilitate particles to move. So this is kind of like they're providing little tunnels that will allow things that can't go through the membrane to go through the tunnel. And you couldn't walk through a hillside, but if there were a tunnel through the hillside, you could go. Culverts under your driveway, the water would not just go through the rock or the yard in your driveway, but if there's a culvert or a tunnel, it will go through. So these are providing places. They are specific for each different substance. The sodium can only go through the sodium channel. It's just the right size and shape for a sodium ion. Calcium can only go through calcium. Glucose and amino acids have their own channels. So if a calcium tried to go through an amino acid channel, it would not be able to fit. So these are just little passageways in a cell membrane. That allow things to go in and out. This is one of, these three things are one of the things you'll be describing in your first 3 bullet points in your introduction. I expect you to have done by the time you show up tomorrow. That way we'll spread this work out and you won't be stuck trying to write the whole thing on Sunday night. Don't be that person. A little bit today, a little bit more tomorrow. If you show up on Wednesday with no procedure, it's going to be pretty tough to do the lab. So that's what our tomorrow is for, to make sure you have some time to work with that and you understand what you're doing. Third kind is where we look at a carrier protein. And again, we have the inside and the outside of the cell. In the carrier protein, we have an amino acid, which might be this green thing, okay? The amino acid. And it attaches to a receptor. The receptor then opens the channel and lets that particle go through. If the right amino acid attaches here, the channel opens. If something else that tries to attach there, like a different amino acid, or maybe a lipid or something else, a steroid, a hormone, a chunk of who knows what, and attaches to the receptor will go, no, you can't come in, will not open. The right thing, the channel opens and the amino acid can go right through into the cell. So this is the carrier protein. Again, it requires no energy. It's just a little chemical reaction. Think of the particles knocking on the door and then the door opens. They do the right secret code knock. In all three of these processes, there is no energy spent by the cell. The process is driven completely by the concentration gradient.